All right. Hi, my name is Sophie Shen. I'm from Richfield High School, and this is my research project on the efficacy of curcumin as a potential therapeutic agent in tauopathies using Drosophila melanogaster. So, as a quick background, unfortunately, around 2022, my grandfather was diagnosed with Alzheimer's disease. And this actually coincided with around when I joined the research program at my high school, and I thought this would be a great opportunity to kind of understand what he was going through and learn more about what he was experiencing. And so through my research, I learned two key facts. First, that an estimated 22% of people older than 50 are on the Alzheimer's disease or AD continuum, meaning like early, mild, and late stage symptoms of Alzheimer's disease, and that's around 460 million people. Additionally, current FDA-approved drugs, there are six, for the treatment of AD are limited because they're more symptomatic treatments, not disease-modifying, meaning that they can only temporarily address symptoms, but not slow down the progression of the disease. So now onto tauopathies. Tauopathies are a class of neurodegenerative disorder characterized by tau aggregation. So as you can see in this image of a neuron, your microtubules are the green lines depicted. And so in so the microtubules are important for intracellular transportation and maintenance of cell shape. And what healthy tau protein does is it stabilizes the microtubules. However, in the bottom image, as you can see in tauopathies, researchers don't know exactly why, but tau hyperphosphorylates, causing it to destabilize the microtubules. And so the tau monomers, they clump into oligomers and then neurofibrillary tangles, which this causes cell death and causes the neuronal dysfunction like cognitive and motor impairments that you see in AD and many other um, tauopathies. So the reason why I focused my research on tauopathies instead of AD, my initial interest, is because after talking with and like emailing a lot of the authors of the journal articles I was reading, they recommended that because I was using invertebrate model organisms, that it would be a lot more efficient to focus on tauopathies instead of AD, because AD is caused not only by tau protein, but also amyloid beta, and tracking both of these factors would be a lot more difficult. Moving on to curcumin. So I mentioned earlier that current FDA-approved drugs for the treatment of AD are limited. And so in the absence of effective disease-modifying um, remedies, a lot of things like plant extracts have garnered attention because they have research studies have showed that they have um, promising like therapeutic effects, but a lot of them are under-researched. So one of these under-researched plant extracts that attracted my attention is curcumin, which is a compound derived from turmeric, and it's demonstrated disruption of amyloids in AD and therapeutic potential in chronic inflammatory diseases. So these factors kind of made me want to look into curcumin more. So at this point in my research, I'd identified two main problems that I saw in the field of tauopathy research. First, that current drugs treating tauopathies are limited, failing to slow down disease progression. Second, many promising treatments, such as curcumin, are under-researched. Then this led me to my research objective, to assess the efficacy of curcumin in slowing tauopathy progression. And so how would I do this? I decided to use Drosophila melanogaster, aka fruit flies. So, fruit flies are very valuable in the field of telepathy research because they're highly genetically tractable, they have a small size, they're inexpensive, and they have a short lifespan. And so, moreover, more important to me was that the inside skin from Drosophila are highly relevant to higher level species, including humans. And that's because over 75% of genes implicated in human diseases have Drosophila orthologs or like analogous genes. Additionally, this is really important for our research. Human tau expression in Drosophila causes locomotive defects in both adult flies and larvae, and I'll touch more upon that later. So you might be wondering at this point, human tau expression in Drosophila, like how does that work? How do you express tau, human tau, in fruit flies? And so in my research, I use the Gal4 UAS system. Basically explaining this process, you have a Gal4 line of the fly, and that fly has a Gal4 gene that codes for the Gal4 protein. That fly also has a tissue-specific promoter. Here it's the INSIP promoter, and this promoter activates the Gal4, resulting in the protein. And then in the UAS line, the UAS is only activated if bonded with gal4 protein, and that UAS codes for tau. So normally this fly doesn't express tau. However, when you cross it with a gal4 and you get this line of fly, it has both gal4 and UAS, and the UAS bonded with the gal4 protein triggers the expression of the reporter gene. Here, that's tau. So that's how you get a tau expressing fruit fly. Now moving on to my methodology. In my research, I had two wild type like control fly stocks, the ATTP2, which is a genetic background, and W1118, which is sort of a standard wild type reference. The genetic background was used to create the tau expressing lines, GFP, EGFP, and 2 and 4 are tau. The main difference between these lines is just they have different fluorescent tags employed for labeling and live imaging, but what was important to me is that they're all tau expressing. For the fly husbandry, fly stocks were maintained on standard food at 24 plus minus 1 degree Celsius temperature and in a natural light cycle. This was in May 2023 in Connecticut. For the fly food, I cooked it as previously described and in a previous journal article with 1% agar, 5% sucrose, 5% dry yeast, and 3% powdered milk. And you can kind of see like the fly stalks in these images. To treat the fruit flies with curcumin, curcumin was added to the standard food at a concentration of 0.037% at 55 degrees Celsius, and then I also kept it out of direct sunlight to prevent photooxidation. I'm going back to this image. So for each fly stock, I had two groups. One group was treated with curcumin, and one group was just raised on the regular fly food without curcumin. So in total, I had 10 groups. Flies, yeah, and then so I fed the flies the curcumin and food combination in order to treat them with the curcumin. Now, the larval crawling assay. So I've mentioned previously that human tau expression in Drosophila causes locomotor defects in both adult flies and larvae. And this fact was very important for my research because I used the locomotive defects, which is like movement. I used movement as a measure of disease severity in my fruit flies. So much like how dementia and neurodegenerative like decline is a symptom of tauopathies in humans, and you can use that to see how severe the tauopathy disease is in a human. In the fruit flies, I measured and looked at movement to see how severe the tau expression and the progression of disease, how severe that was in my fruit flies. In my larval crawling assay, I used third instar larvae. I used these larvae to make sure that all my larvae were at the same age. And at this stage, the larvae are very easy to recognize because normally in this image, you can see larvae usually stay in the fly food. However, when they're at this stage, they'll crawl up to the sides of the fly vial, making them easy to recognize. And then I put these larvae onto a petri dish that was over a grid paper. I let them acclimate to the petri dish for 30 seconds, and then I recorded their movements for 1 minute. And then I imported these 1 minute videos to Fiji, which is a video analysis software, and I used the MTrackJ plugin to manually track the movement for the larvae in each video. I tracked where their head was every 2 frames. And I used GraphPad Prism 10 for statistical analysis. I did a one-way ANOVA and then a Fisher's LSD test. So now my results. First, curcumin treatment improved larval crawling and tau expressing larvae. So my ANOVA test showed that there was a significant difference among mean distances. I got a p-value of 0.03. So p-value shows statistical significance. If you get a p-value below 0.05, that shows that the results were statistically significant and likely not caused by random chance. Additionally, the mean recovery, which is the difference between the average distance between curcumin treated larvae and not treated larvae not treated with curcumin, and so this was the mean recovery for each tau stock line, 0.81 centimeters, around 2 centimeters, and around 1.5. And so for two of these stock lines, EGFP and 2N4R, I found that there was a statistically significant difference in the recovery. I got a p-value of 0.004 and a p-value of 0.03.
Hey, Sophie, you
As
have
you can
just over
see,
2 minutes left.
Thank you, as you can see in this graph. Okay, and then another important, sorry, oh wow. Okay, going back. So this showed that curcumin, um, oh, was this statistically effective in recovering the locomotive the movement in the larvae. Another really important result that I found was that curcumin treatment did not affect larval crawling in control larvae. So as you can see, in the p-values for both controls, there was not a there is not a significant difference in mean distance traveled. So this told me two things. That first, curcumin didn't cause an adverse effect in the control larvae. But more importantly, it showed me that curcumin didn't act as a general stimulant. For example, how like coffee might act as a general stimulant for us. It showed that because it didn't like increase the movement in the control larvae, that the curcumin directly targeted something in the tau pathways of the tau expressing fruit flies, which is what rescued the larval movement, not like an overall boost of all of the larvae. So conclusion, my main takeaways in my research are that I found statistically significant evidence for curcumin's ability to mitigate locomotor impairments induced by tau protein. Some of the remaining problems after my research is I'm still not quite sure why the GFP tau line didn't yield significant results. Also, there's emerging studies demonstrating that curcumin has limited bioavailability, meaning that for humans it's difficult to absorb, which may uh, be a roadblock in using it as a therapeutic agent. So in my future work, what I hope to do is scale up by doing more trials and also testing curcumin at various concentrations to see an ideal concentration. Thank you so much. Thank you everyone for being here and listening to my research. I really appreciate this.